Welcome back. This is our second segment on gene expression. Last time we looked at the fundamental differences between the execution of gene expression in prokaryotes and eukaryotes. And so we're going to start this time by taking a look at the details of transcription. So transcription, remember, is the reading of information in a gene, sequence of nucleotides that's going to produce a functional product, such as a tRNA, an mRNA, or ribosomal RNA. Numerous other types of RNA as well. So transcription is this reading of information in a gene to produce a functional product. And so we can break the process of transcription down into three general events. Now we're going to focus on transcription of genes which produces RNA. These are so-called structural genes. And so we start transcription at a region of the gene called the promoter. So let's get a pen up here. So here is the promoter. It's this blue blocks here. And so that is the place where transcription will initiate. So transcription is done by a large molecular machine called RNA polymerase. Now there are several RNA polymerases in cells. Commonly we call them RNA polymerase 1, 2, and 3. And we're not going to worry about those right now. But those polymerases, those enzymes are capable of joining nucleotides together using the information in the gene sequence. So if there's a C in the gene, then the RNA polymerase will read that and say, okay, I need to put a G in my transcript, which is the molecule it will be making. And so the usual base pairing rules apply, except there is no thymine in RNA, as you know. And so instead of putting a thymine in, RNA polymerase will use a uracil. And so if there's an adenine in the sequence that's being read in the gene, then that will correspond to a uracil in the RNA. So this all starts at a region called the promoter, and that signals the start of transcription. So there's a region of nucleotides that the polymerase can recognize, and it recognizes that and grabs hold of that sequence and binds to the gene, and that tells the enzyme, this is where you start transcribing. Start reading the information in the gene here. So that's the promoter. Once then the promoter, the polymerase has started making a sequence of nucleotides using the information in a gene, it's enters what's called the transcribed region. And so we have initiation of transcription of the promoter and then elongation through the transcribed region where the information is just being read in the gene and the corresponding bases, the corresponding nucleotides are being added to the mRNA or whatever molecule is being produced, whichever nucleic acid, whichever RNA is being produced. Then at the other end, the non-promoter end, there's a sequence of nucleotides called the terminator. You don't need to worry about how it does this right now, but the terminator tells the polymerase, this is the end of the gene, you can stop transcribing here and let go of the gene. So we bind at the promoter, read through the transcribed region during elongation, and then we terminate, we stop the process, we let go of the gene at the terminator sequence. And so that produces, via transcription, in the case we're looking at here, an mRNA molecule. So let's break that down and look at it in a little bit more detail. Let's look at the simplest case we possibly can, and let's look at what happens in bacteria. So the mechanics of the process are the same, but the specifics vary a little bit. So I don't need to focus on the specifics, I want you to focus on the general mechanism, because that's the same comparing bacteria or prokaryotes in general and eukaryotes. So let's start with initiation. So initiation is occurring over here on the left. Let me grab a pen. So here is that enzyme called RNA polymerase. I'll just trace it around real quick. So here's RNA polymerase, and it is bound to a region of DNA called the promoter, and that sequence of DNA is going to be in here somewhere, and we can't see the bases, and they're not important. But that's where the RNA polymerase grabs hold of the DNA and it knows that it should start transcription there. And the first thing the RNA polymerase does is it pulls the two strands of DNA apart. And so it pulls the two strands of DNA apart, and that exposes the sequence of nucleotides in the strand of DNA that the polymerase will read. So the polymerase then starts reading, and we're into the elongation phase. So now we're into the actual business of transcription. So the RNA polymerase is going to move along the DNA, unwinding the double helix, reading the information. And so if it sees an A, it makes a U in the new piece of RNA. If it sees a C, it puts a G, and it sees a G and it puts a C, and then it sees a T and it puts an A, and it sees an A and it puts a U. So it reads the information in the gene, making this RNA molecule, and here we're actually making an mRNA. Now, if you remember back to our discussion on DNA replication, is what hopefully you remember is that you must always make nucleic acids in the direction of 5' to 3'. And so we're making the new molecule, the RNA molecule, in the direction of 5' to 3'. So that means we're adding new nucleotides onto the 3' end. So this would be the 5' end, and we're adding nucleotides onto the other end, the 3' end. So we make the new nucleic acid, the RNA, in the direction of 5' to 3', but the information in the gene is read in the other direction, 3' to 5'. And this goes back to our whole discussion of anti-parallel, of things that are anti-parallel when we talked about DNA structure. So we read the information 3 to 5, but we make the RNA that we're building 5' to 3'. And so we can see that in this box here. So during elongation, the DNA template strand, that's the one that contains the information. So that's this part up here. The DNA template strand, that contains the information, is being read as we see it here from left to right, but chemically, that would be from, see if I can do a 3. That would be from 3', it's nearly a 3, it's good enough for us, I think. So that would be the 3' end, and then this would be the 5' end if I can make a 5. So that's the direction we're reading the information. That's in the DNA template. But we're making the RNA in the other direction. We're making it 5' to 3'. So that follows this rule that we always must make nucleic acids in the 5' to 3' direction. So the RNA polymerase is reading through the gene, and when it sees an A, it makes a U, T, it makes an A, so on. It reads through, and then it gets to the terminator sequence here. And there's something special about this terminator sequence, and we're not going to talk about it, but at the terminator sequence, the polymerase knows to let go. And so it basically lets go of the DNA it's reading, and it lets go of the RNA transcript. And then the RNA transcript, the mRNA molecule in bacteria, can then be directly translated. Now let's take a quick look at some variations in eukaryotes, and these variations are very important. The mechanism of transcription is very, very similar. That is to say that we're always going to have this promoter. So here is our promoter. That's where we're going to start transcription. We've got a transcriber region, and then we've got a terminator at the end. So the general structure of this gene is similar to what we saw in bacteria, but there are going to be some very important differences that we're going to focus on in this slide. So let's look at the first main difference to start with. In the eukaryotic structural gene, remember a structural gene is just a gene that's going to make an mRNA. That's compared to a gene which might make a tRNA, or a ribosomal RNA, or a microRNA. So this is a gene which is ultimately going to lead to the production of a protein. So that's what we mean by structural gene. In eukaryotic genes, there are two parts to the gene structure. Those two parts are exons and introns. And the exons and the introns alternate in eukaryotic genes. And so the whole thing is going to be transcribed, and we'll come back to the exons and the introns in a moment. You've just got to be aware that there are different sequences in the transcribed region. So the whole thing is transcribed. So we're going to read all of this information in here, and we're going to do the process of transcription like we just talked about. There are variations when you compare prokaryotes and eukaryotes, but you're going to get into the details of those in molecular and cellular biology, so let's not worry about them now. We've made our mRNA. Now the first molecule that's made from the gene in a eukaryote is called a pre-mRNA. And so here's its 5' end, and here's its 3' end. And you can see that the two types of sequence that we had in the gene, the exons and the introns, are also present in the pre-mRNA. So this here is exon, it's a pretty bad X, exon 1. This is intron 1. Not a very good one. This is exon 2. This is intron 2. And then this is exon 3. And so that structure that you see in the gene is maintained into the pre-mRNA. So at that point, the pre-mRNA contains these exons and these introns. After this transcription occurring, three processes then occur in eukaryotes: capping, splicing, and tailing. Now we'll look at these in detail in the next segment, but let's just kind of touch on them briefly here. Capping is a process which adds a special nucleotide called a 5' cap to the 5' end of the mRNA. Splicing has taken out these introns. So these intron sequences in the mRNA get deleted. It's like copy and pasting in Microsoft Word, or cutting and pasting in Microsoft Word. You basically take the intron sequence and the cell metaphorically highlights it and then just chops it out, and then it joins the two resulting exons back together. So the end of exon 1 here is joined to the beginning of exon 2 here, and the intron between, this thing gets deleted out, and then this thing gets deleted out. And so all the exons get joined up together to make an mRNA, which is just composed of exons. So that is splicing. We'll look at that in more detail. And the last thing that happens is a process called tailing, and that is where a structure called the 3' poly A tail gets added. And that is just a chain of adenines. So all of these things happen after transcription, so they're said to be post-transcriptional. So we'll talk about these things in more detail in our next segment. That's it for now. I'll wrap up here, and I'll be back soon with our next segment, and we're going to look at splicing, capping, and tailing in more detail.